Hi everyone. This is the third lecture for your chapter Biomolecules. Till last class, we have discussed carbohydrates, we have discussed lipids, and we have also discussed nucleic acids. In this lecture, we will be starting with the fourth and the most important group of biomolecules, which are amino acids and proteins. Clear? Amino acids are the building blocks of proteins. So we will start with the amino acids. As the name suggests, okay, from the name itself you can come to know two things. First, it is an organic compound, okay, that you should know. This organic compound has an amino group and an acid group. That is why they are called amino acids. Clear? Now what you have to know or what you have to remember is this amino acid or amino and acid group are attached to the same carbon. Clear? Let me just write down one structure and you tell me whether it is an amino acid or not. Here you can see a carbon chain. Let us say here it is COOH. And on this carbon I have drawn NH2. Clear? So even this compound, if you talk about, it has one amino group. It has one carboxylic acid group. It should be an amino acid, right? Actually, it is right. Okay, so to specify which type of compound we are talking about, we call the amino acids found in proteins as alpha amino acids. Okay, why alpha amino acids? Because here you can see the NH2, nitrogen has three valencies, right? So this nitrogen has two hydrogen attached to it, covalent bond, and one covalent bond is with this carbon. So this NH2 is amino and COOH is carboxyl. Now both of these groups, they are attached to the same carbon. Okay, and this carbon here is called the alpha carbon. Okay, this carbon is known as alpha carbon. Okay, if you number it, okay, 1 2 3 4 5, then it will be the second carbon. So this is the carbon number 1, this is carbon number 2. Fine. Carboxylic acid carbon will always be given number 1. I think this much you must have understood in chemistry, right? Studied in chemistry. Fine. So whatever amino acids are found in your the proteins, okay, they are all alpha amino type compounds, alpha amino type amino acids. Clear? The particular property of amino acid is its ionizable nature. Okay, ionizable nature meaning it can get ionized and have positive and negative or positive or negative charge. Clear? So let us see here. Okay, now yeah, one more thing. This carbon here, it has one hydrogen group attached to it always, so that one hydrogen will always be there. And on the other side you have R group, a side chain. Okay, that side chain keeps on changing, and with that changing side chain you have different types of amino acids. We shall see that later in this chapter, right? So if you want to write it down, you can just directly write CH, then COO minus, because COOH, carboxylic acid group, can give one proton. Okay, it will give one H plus and become negatively charged. So COO minus. The NH2, okay, the amino group, it can take up one proton, H plus, and it can become NH3 plus. Clear? So this here, it can exist as a Zwitter ion. Zwei in Germany, they call two, okay, as Zwei. In German language, if you want to say two, you say Zwei. So from that Zwei comes this name Zwitter ion. Okay, it means two ions. Which two ions? This positive one and this negative one. Clear? So on the same molecule, on the same amino acid, you can have two charges, positive as well as negative. Clear? With change in pH, the structure and ionization of amino acid changes. Okay, let us say you have acidic pH. Acidic pH meaning in the solution you have more amount of H plus. Okay, there are more protons in the solution. In this case, what will happen? This COOH group, okay, whatever the amino acid has carboxylic acid group, it will not release its hydrogen. So this COO minus will be in COOH form only. It will take up that proton from the solution, or it will not release its proton only. Clear? And on the other hand, the amino group, it will take up a proton because there is a lot of protons in the acidic pH, in the acidic solution. So it will take this one proton and become NH3 plus. So in acidic pH, when there are many protons, many H plus outside, the amino acid will try and take up that H plus. When the COO minus takes that H plus, it becomes COOH. And when the NH2 takes that H plus, it becomes NH3 plus. Overall, if you see the charge on this amino acid, it is positively charged. Clear? So in acidic pH, the amino acids are positively charged. The opposite will happen in case of basic pH. Now in basic pH or in alkaline solution, you have less amount of H plus in the solution. So what will happen? This NH3 plus will give its proton. Okay, the H plus will be released and it will become NH2. Same thing with the COOH, okay, the carboxylic acid group. It will also release its proton, and so overall charge on this amino acid will be negative. Okay, the COOH will be in COO minus form. So this is the special peculiar property of amino acid, that it can be ionized. Okay, it can act as a Zwitter ion, which means it has positive as well as negative charge, and with differing pH. Okay, in acidic pH and basic pH, it is positively charged and negatively charged respectively. There are 20 different amino acids which occur naturally in the proteins. Fine. And the last thing I want to tell you about the structure of amino acids is, remember in carbohydrates we studied two isomers, L isomer and D isomer. I told you all the sugars in your body are D type. Okay, you have D glucose, D glyceraldehyde, D fructose. D means dextrorotatory, which means they shift the plane polarized light towards the right side. Okay, clockwise direction. Now think of this molecule. Okay, think of this amino acid. Is this carbon also chiral carbon? Right? Because it has one valency as hydrogen H, one valency is COO minus, other valency is NH3 plus, and the fourth valency, the R group, is again another different group, right? So the definition of chiral carbon or asymmetric carbon is all of its four valencies are filled with different functional groups. Is it true here also, right, in amino acids? Now, in carbohydrates, we considered the last fifth carbon or whatever the second last carbon will be there. Okay, whether the OH group, hydroxyl group, was on right, then it would be D. If it is on left, it would be L type, right? Same way, in case of amino acid, we consider the amino group. If it is on the left, we call it L amino acid. If it will be on the right, we will call it D amino acid. Like how sugars are always in D form inside your body, the amino acids are always in L form, which means the NH2 or NH3 plus will always be on the left side of the amino acids found in the proteins and found in your body. Clear this much? So if somebody tells you about the optical isomerism or just isomerism, okay, what kind of isomer is the amino acid? It will be D or L type, and mostly what is seen in nature is L type of amino acids. Clear? Right. So now there are 20 different alpha amino acids which occur naturally in the proteins. We shall be seeing all of those 20 one by one. Why these structures are important, or why understanding the structures are important, is because based on this, a lot of different questions can be asked. If you know the structure, or if you have at least understood the structure, you will be able to answer a lot of questions. Okay, so let us start. This will be the basic structure of amino acid. Okay, you have NH2 on the left hand side because it is an L amino acid. Then you have CH, okay, and COOH on the right hand side. At the bottom, we will write the R group. Clear? This will be the basic structure of amino acid. Let us start with the most basic amino acid, which is glycine. Glycine has R group H. So, can you see something here? NH2, COOH, the same thing. But this carbon has one hydrogen on one valency and another hydrogen on another valency. So, this is not asymmetric carbon. This is not a chiral carbon. So, glycine is an exception. Glycine is the only amino acid which is not optically active. Clear? I hope you are noting all of this down. Clear? Fine. So, the simplest amino acid is glycine and it has H functional group. Okay, and R group will be nothing but a simple hydrogen atom. Why it is called glycine? Because when you taste this amino acid, it tastes sweet. Okay, so because it tastes sweet, people used to think that it must be a carbohydrate. Later on, they found out it is not a carbohydrate, it is an amino acid. So they named it glycine because of its sweet taste. After H, the second simplest R group is methyl group, CH3. If you attach methyl group on the R, this bond, okay, as the R group, it is called alanine. Alanine is optically active. As you can see, the four different R groups, okay, four different functional groups are seen here, okay, attached to the carbon. So alanine is again optically active. After alanine, which is the simplest CH3, okay, as R group, we have the next one, which is valine. Now here I was not able to write it like a V, but this is how I normally draw it. Okay, this R group, if you want to draw, you start with this C and then you draw it like an inverted V. Can you see this? This would be your normal V. Draw it upside down. Okay, and you get this structure of valine. Now, if you see this carbon, it already has 3 valencies covered. So you just write CH and these both will be CH3, CH3. Clear? So this is how you remember valine. Valine, wherever you hear, it has that V shape. And with V shape, each end will have one carbon. So you have 1, 2, and 3. Just fill up the valencies and you know how valine looks like. Clear? Fine. After that, we go to the next simplest amino acid, which is leucine. Okay, we'll start with leucine. Leucine is same as valine, except it has one more carbon. Okay. Right? Remember valine? We just saw it right now. Valine is this inverted V shape. It has 3 carbons, 1, 2, and 3. Now add one more carbon to this chain. Okay, make it an inverted Y. This inverted Y is nothing but leucine. Okay, and this Y here there will be one C, 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 and C. So it has 4 carbons in its R group. Okay, just fill up the valencies. So you have CH2, CH, and CH3, CH3. So the same thing. You have the valine, and with that valine, an added CH2 group. Okay, so instead of V shape in valine, leucine is Y shaped. Clear this much? I hope you will be able to remember now. Okay. See, sometimes it comes with practice. So what I want you to do is write down with me, and after that, just go through all of these. Then close your book, take a paper, and try and draw whatever you can remember. Whichever amino acids you do not remember, just refer to those amino acids in the book and then try to draw them. Okay, attempt to draw them. This way you will become more and more capable of remembering all these R groups. Clear? Then you have isoleucine. Leucine, as we saw, has 4 carbon. Okay, 1, 2, 3, and 4. Isoleucine also has 4 carbon. Have you studied different isomers like iso, neo, right? So iso means what? It has the same 4 carbons, but instead of a Y shape, it will have something like a L shape. Okay. So this first carbon, CH, will get one methyl group, and the other 2 carbons, CH2, CH2, will be arranged like this. Okay. Just like leucine, this also has 4 carbons, but the carbons are arranged a little differently. Isoleucine meaning the first ever carbon of the R group will have a CH3 group, and on the other hand it will have a ethyl group. Clear? Then you have methionine. Methionine, if you see, is also same as leucine or isoleucine. It also has 4 what you can say members, okay, in the R group, except instead of 4 carbons, it has 3 carbons and 1 sulfur. See, this is why it is important. Methionine has a sulfur atom. Sulfur has 2 valencies. So just remember the R group. Here it will be CH2, CH2, S, CH3. So there is a methyl group attached to the sulfur, and that sulfur is attached to this CH2, CH2 chain. Clear? So this is methionine. Methionine, once again, I'll quickly revise for you. It is a sulfur containing amino acid. And second thing, when you study the translation, okay, whenever you will study molecular basis of inheritance, you will study translation. So during transcription and translation, the first codon, the first message is always AUG, and AUG stands for methionine. So most of the proteins will have methionine as the first amino acid. Clear? Then you have phenylalanine. Now do you remember alanine? It was simple, right? CH, and this R group had CH3. Clear? Simple methyl group. Now look at the name. It has phenylalanine. This ring structure is nothing but your benzene ring. So if you attach a benzene ring to some molecule, you can call that molecule phenyl something. Okay. In this case, it is attached to the alanine. So you call it phenylalanine. Clear? So phenylalanine is the first aromatic amino acid we will study. Okay, phenyl, this is the phenyl ring, okay, which is nothing but a benzene ring, and this benzene ring is attached to the CH3 or CH2 group of alanine. So it is called phenylalanine. Now to this phenylalanine, okay, you just add one OH on the opposite side, on this side, and it becomes tyrosine. Clear? Same thing, you draw alanine. From that alanine, you add one phenyl ring, it becomes phenylalanine. To that phenylalanine, you add OH, okay, one hydroxyl group, and it becomes tyrosine. Tyrosine is an amino acid found abundantly in cheese. Okay, it is found in cheese, and it has that cheesy smell. When you know how fermented cheese smells like, it is the smell of tyrosine. Clear? So here, this is the structure of tyrosine. It is just phenylalanine with one OH group. So remember this. This is an aromatic amino acid, phenylalanine. Tyrosine is also an aromatic amino acid, but it is also the first amino acid we have seen which has hydroxyl group in the R group. Okay, so it has this hydroxyl group, OH group, alcohol group. Now let us talk about serine. First of all, let us draw the alanine. Okay, so here CH3. Now take that one hydrogen and replace it with hydroxyl group, OH group, alcoholic group, right? So it becomes CH2OH. The CH2OH derived from alanine, right? Very simple. Now we will be able to understand. Remember, right? So CH2OH, this is the second amino acid we studied which has OH group. Clear? And then we talk about the third amino acid. This is just like valine, but instead of 3 carbon, it has 2 carbons and 1 oxygen. Okay, so it has CH, CH3, and on the CH, the second carbon, it has OH group, hydroxyl group. So tyrosine, serine, and threonine. These are the 3 amino acids which have hydroxyl group or which have alcohol functional groups. Clear? Tyrosine, easy to remember, it is just phenylalanine with an OH group. Serine is just alanine with an OH group. Threonine is just like valine, but instead of in that V, we were writing CH and then CH3, we write CH and OH CH3. Clear? So these two groups and you get this threonine. Fine. Followed by that, simple. Again, draw that alanine, which is CH3, remove one hydrogen and add a sulfhydryl group. Remember this name, SH is the sulfhydryl group. Okay, so when you attach this sulfur, and sulfur, as I told you, has 2 valencies. So one, it is bound to the carbon, and the other one is with hydrogen. This is the second amino acid we studied, which has sulfur. The first one was methionine, and the second one is cysteine. Clear? Remember this cysteine because in proteins, this cysteine is what helps in the disulfide bond. Okay, we will see that later. But this SH group is very, very important. Even in metabolism, I will keep on referring to this SH group. Okay, it is your sulfhydryl group. Just like OH, which is your alcohol group, this is sulfhydryl group. Then you have the third aromatic amino acid, which is histidine. Histidine is again just like alanine, you have CH3, replace that hydrogen with this group. How I remember it as is, you draw a pentagon. Okay, you draw this pentagon. It looks like a house if you look at it from side. Let us say this is the house, it has this entrance, right? And at the ends, okay, here, let us say there are windows. Fine. These windows are the places where nitrogen is present. Clear? So histidine has this heterocyclic ring. Heterocyclic meaning it has carbon as well as nitrogen in its ring. And what is its structure? It is this pentagon ring. Okay, it has this 5-membered ring. And if you think of it as a house, then wherever there are windows, which is these 2 points, there nitrogens are present. So this is your histidine. This is the third aromatic amino acid. Then you have aspartate or aspartic acid. Aspartate, normally you call it when the COOH is in COO minus form. Okay, when it is in deprotonated form or when the H plus has been removed, then you call it aspartate. If that COOH is just present like this, you call it aspartic acid. Clear? It is one and the same molecule. Fine. So again, you start with alanine, you have CH3, replace that third hydrogen with a carboxylic acid group. So you have CH2COOH, and this is aspartate. As you can see, this is acidic R group. Okay, so this amino acid will be acidic in nature. Same as aspartate is another amino acid, asparagine. This COOH, carboxylic acid, when it reacts with ammonia, okay, what it gives CONH. This CONH, okay, so CH2CONH is asparagine. Clear? This is called amide. CONH is called amide. Right. After aspartate, you have another amino acid, same as aspartate. What is different? You have one more CH2. Okay, the chain length is different. CH2COOH is aspartate. CH2CH2COOH is glutamate. Clear? Only one CH2 is extra. So this is again glutamic acid if COOH form is present and glutamate if COO minus form is present. From this COOH, if it reacts with ammonia, Okay, acid plus ammonia, which is basic in nature. You get CONH, amide. Clear? So this here is glutamine. Same as asparagine, you have glutamine from glutamate. Fine. Now, talking about some complicated amino acids. Simple, but a little bit difficult to remember. Here, you have lysine. See, we saw the acidic amino acids, right? Aspartate and glutamate. They have acid group in the R functional group, right? Here, this is the basic amino acid structure, and in its R group, you have CH2 whole 4 times. So there will be 4 times CH2, 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 CH2. Clear? And the last CH2 will have one ammonium group, NH3 plus. Clear? So this NH3 plus gives it a basic nature, which means lysine is basic amino acid. Then we have arginine. Arginine is again the same way, CH2 whole thrice. So you have 3 times CH2, CH2, CH2. After which there is this special kind of functional group. Okay, can you see? You have NH, okay, so normal nitrogen, and with its with it is one hydrogen, so 3 valencies, NH, then one carbon. To this carbon, 2 more NH2 groups are present. Clear? So just write it like this, or remember it like this. It has 3 NH groups, okay, or NH2 groups attached to it. Clear? So this is nothing but the structure of arginine. You do not have theory exams, so you do not need to draw these. But why I am explaining is, if you have seen the structure, you will be able to answer a lot of questions. Remember this glutamate, glutamine, asparagine, because even when we will be discussing the nitrogen metabolism, okay, in that nitrogen metabolism, we will see the transfer of nitrogen, okay, the transport of nitrogen happens in form of amides, like this. Clear? So these are important in such manner. Fine. So arginine and lysine, both of these are positively charged or basic amino acids. Then you have proline. Proline is very different from other amino acid. See, it has CH, and then R group has the CH2, CH2 and CH2. But instead of it being CH3, that last carbon is attached to the nitrogen, okay, of the same amino acid. So it again forms this kind of a structure, okay, a house-like structure, a pentagon-like structure, and the R group is connected or is has covalent bond with its own ammonia group, okay, NH2 group. So this kind of structure is of proline. It is not an amino acid, it is an amino acid. Okay, the group formed here, which will be NH. Okay, it will not be NH2, it is my mistake. It will be NH, and this NH will have one valency with the carbon and one valency with this carbon of R group. So this is called amino group. Okay, which is why this is not an amino acid, it is an amino acid. Clear? And finally, the last amino acid you have to study is also an aromatic amino acid, and it is called tryptophan. Tryptophan, as you can see, has this kind of a structure. It has a hexagon ring and a pentagon ring with one N, one nitrogen, okay, in its ring. So this tryptophan is also aromatic amino acid. And now I hope it is clear to all the structures of all the different 20 amino acids, what they are made up of, okay, the R groups specifically. And what I want you to do is try and, as I told you, write these down on your own. Fine? Okay. So this is classification of the amino acids based on their R group. Okay, based on their R group. You can have non-polar meaning not having any charge, not complete charge, nor even the partial charges. Okay, so they are non-polar and have aliphatic R groups. Aliphatic meaning linear chain carbon R group. Okay, so it includes lysine, which is just hydrogen, alanine, which is CH3, valine, which is that V-shaped, okay, aliphatic chain, leucine, Y-shaped, methionine, and isoleucine. All of these are non-polar. They are not polar. Then you have polar but uncharged R groups. So they are polar. They have some partial positive, partial negative charge, but they do not have any positive or negative charges. Clear? So there is serine. Here, because of oxygen, oxygen will try and attract electrons from the hydrogen, which is why that oxygen is partially negative and that hydrogen is partially positive, which is why this group, OH group, will always be polar, right? So serine, threonine, they are polar amino acids. Then cysteine, cysteine has that SH group. Sulfur also does the same as oxygen. So SH group is also polar. Proline is also polar. Then you have asparagine, which is CONH, and glutamine, which again has that CONH. Here, so all of these amino acids are polar, but they are not charged. Then aromatic R groups, you have phenylalanine, tyrosine, and tryptophan. Only three. Remember these names. Positively charged R groups. Okay, there is some positive charge. It includes lysine, arginine, and histidine. Okay, these three have plus charge in the R group. And then negatively charged R groups are the ones which are acidic. So aspartic acid and glutamic acid. So this is how, based on whether they are positively charged, negatively charged, non-polar, polar, this is how you can classify the amino acids. They are also classified nutritionally in these three types. Okay, so you can have essential, semi-essential, and non-essential amino acids. Essential means you cannot produce them in your body. Okay, because you cannot synthesize these amino acids. You have to take them in your food, in your diet. Such kind of amino acids are histidine, lysine, tryptophan, phenylalanine, methionine, threonine, leucine, isoleucine, and valine. Then there are non-essential. Non-essential are easily made in your body. Example are glutamic acid, aspartic acid, alanine, proline, hydroxyproline, and cysteine. Okay, these are very easily made in your body, and so you do not require them in food. Then there are semi-essential amino acids. These are not required in higher quantity. Okay, and somewhat your body can salvage, but it does require these amino acids from the diet. So semi-essential amino acids are arginine, tyrosine, cysteine, glycine, and serine. Clear? So nutritionally you can classify amino acid in these three categories. Now let us come to formation of a dipeptide. We have understood the structure of amino acid. We have discussed all the different R groups and the 20 different types of amino acid, how they are classified. Now we talk about the formation of dipeptide. How we have already discussed structure of disaccharide. So same way we will be seeing dipeptide. As you can see here, you have amino acid number 1 and amino acid number 2. Here amino acid number 1 has carboxylic group and amino acid number 2 has amino group towards each other. What will happen is this hydroxyl group of carboxylic acid OH and this H, they will react. Okay, the water will be removed. This OH and H will be removed as H2O. So the carbon and nitrogen will form a bond. This bond is called peptide bond. Okay, it is CONH. The CO, oxygen, and the hydrogen will always be on the opposite side. Okay, so it will always look like a chair, like this, like number 4. Clear? So it will be CONH. The CONH is peptide bond. Okay, one more thing, if it comes anywhere, this peptide bond is partial double bond. Okay, it has this partial double bond nature because this C, it has partial double bond with oxygen and partial double bond with nitrogen. It is it has a partial double bond nature. Okay, fine. So after studying about amino acid and dipeptides, we come to proteins or polypeptide. Polymer of amino acid are called polypeptides. So proteins are polymers or polypeptides. They are linear, okay, in a single line. They are not branched. You do not have any branching from one part. Let us say some other amino acids will start forming a line. No, they are a single line, okay, of polymer. It is heteropolymer. Heteropolymer because it can have 20 different types of monomers. Clear? So it is a linear heteropolymer of amino acids linked by peptide bonds. Now, just some facts. Collagen is the most abundant animal protein. Collagen, as you must have studied, is found in your extracellular matrix, okay, in your tissue. Then it is found in your bones, it is found in your skin. So collagen is the most abundant animal protein. And rubisco is most abundant plant protein. Ribulose, bisphosphate, carboxylase, oxygenase. Okay, that enzyme which we will study in the next chapter, which is photosynthesis. Rubisco is enzyme which helps in this photosynthesis process. Clear? It is an enzyme. So collagen is abundant in animals, rubisco is abundant in plants. Here is a table from your textbook of different proteins and where they are found. So collagen is intercellular ground substance, which means between two cells and underneath those cells. Have you ever heard of the term basement membrane? Right? That basement membrane also has a lot of collagen. Clear? Some support material on which the cells can be present. Okay. Then you have trypsin. This you would have studied in your pancreas or digestion, right? You have enzyme trypsin, which is secreted to digest proteins. Then insulin. Insulin is an hormone which is secreted by your pancreas. Then antibodies in immunology, you must have read about it. Antibodies are nothing but they are um, Y-shaped proteins. Okay, they have heavy chain, light chain, right? And they help fight infectious agents. They help fight infections. Then you have receptors. Receptors are nothing but they are proteins which are present on your cell membrane, right? So you can have sensory reception like smell, taste, then receptors for hormones, neurotransmitters, all of these are nothing but proteins. And then finally, GLUT4 is glucose transporter type 4. This glucose transporter is a channel, okay? It is a channel protein found in your cell membrane. And this GLUT4 allows glucose, which is polar molecule, right? So it is polar molecule and it cannot pass through the cell membrane. So it passes through this GLUT channel, okay? GLUT, which is glucose transporter type 4. So it enables glucose transport into the cells. Clear? Now talking about the structure of protein, it can be divided into 4 different stages. The first stage is primary structure. Try and remember translation, which is from your mRNA, a polypeptide chain, okay, and what you can say string of amino acid is formed, right? So this is called its primary structure. Okay, it will have an N terminal, the amino acid which is first. It will have the ammonia group, okay, NH3 plus group, which is free. So this means this will be its N terminal, nitrogen terminal. Then there will be a long line of different amino acids connected with each other through peptide bond. Okay, and the last amino acid, which will be the C terminal amino acid because it has carboxylic acid group free. Clear? So first amino acid will have NH3 group free. Last amino acid will have COO minus group free. So it is called N terminal and this is called C terminal. And this string, okay, this chain of amino acids is called peptide. Okay. So it is a linear polypeptide. Fine, polypeptide meaning it has a lot of peptide bonds. The first amino acid is also called N-terminal amino acid. Last amino acid is C-terminal amino acid. A protein thread does not exist as a extended rigid rod. Okay, this is more like a thread. Okay, this polypeptide will be like a thread. It is movable. It is not like a rigid rod. Okay, it is not like a pencil or a pen in a straight line. It is more like wire. Okay, it can move in any direction. After the primary structure comes secondary structure. So, some amino acids can form hydrogen bonds. Okay, remember the amino acid which had OH group, okay, which were polar. So, such kind of amino acid can form hydrogen bond. The R groups can form hydrogen bond with each other. So, in this case, the amino acids will form this kind of um, secondary structures, okay, which are known as alpha helix or beta pleated sheets. Here, the nitrogen and the oxygen of the functional groups, okay, NH3 and the carboxylic acid group, both of these will also help in formation of this hydrogen bond. As you can see here, this is a helical shape, okay, it is a helix, like a spiral, like a spring, and the dotted lines are showing nothing but the hydrogen bond. Clear? So because some amino acids can form hydrogen bond with each other, it will form this sort of alpha helix. Then they can also form secondary structure like this, which are beta pleated sheets. Have you ever seen those aluminium or I don't know, iron sheets? You normally call them, I think in vernacular, chapra, right? Metal na chapra joyu hoy. If you have seen those kind of metals, they have this kind of shape. Okay, let me just try and draw. They have this sort of shape when you look at them from the side, right? So this kind of a shape, if you imagine, okay, of amino acids, it is forming this beta pleated sheets. Here, as you can see here, and between these two sheets there will be hydrogen bonds which are forming here, 1 2 3 4. So this is how the hydrogen bonds will form. How this happens is, remember you had that polypeptide. So this is that polypeptide, okay, the N terminal and the C terminal. Now in secondary structure, what will happen is, from this there will be folding, okay, like this. So now you have N terminal here and C terminal here. These are the beta pleated sheets, and in between these two there will be hydrogen bonds. Clear? So this is how beta pleated sheets are formed, and they are the secondary structures. Clear? After secondary structure, you have tertiary structure. Okay, one more thing. In some textbooks or some books where you might be referring, they write primary like this. Okay, 1 and this degree symbol. That is primary. This is secondary. This is tertiary. Okay, this is quaternary. They mean one and the same thing. So if there is any question where they mention this kind of nomenclature, this means secondary, tertiary, and quaternary. Right. Coming back to tertiary structure. So a single polypeptide chain, which now has alpha helices and beta sheets, they come together to form 3D structure. Alpha helix and beta sheets, they now have some this polypeptide in between, and all of them will come together to form a structure like this. Let me just draw. Okay, let us say this is your alpha helix, and then there is some free chain, free polypeptide, and then there is this beta sheet. Okay, and then it is folded like this. So this will be in 3D. Okay, so there will be a three-dimensional structure to it, and this is called nothing but the tertiary structure. It looks more like a ball, okay, or it has some specific shape. Here in this structure, in tertiary structure, you have ionic bond, okay, Remember the positively charged amino acids were lysine, histidine, and arginine. So you can have those R groups forming an ionic bond with the negatively charged, which were aspartate and glutamate. Clear? Then remember the disulfide bond. Okay, the cysteine. Cysteine has SH sulfhydryl group. So let us say in the same chain there is another cysteine which also has sulfhydryl group. Clear? Let me draw it like this. This is the polypeptide chain and it has sulfhydryl groups, okay, because of cysteine. What will happen is these hydrogens will be removed and there will be the sulfur-sulfur bond, which is called disulfide bond, which is formed. Clear? So here what you are able to see, it is a covalent bond, okay, between two sulfurs. Then there could be hydrogen bond between different partially negatively charged oxygen and between this partially positively charged protons. Here, hydrogen. Let me just clear this up so it looks less dirty. Fine. Okay. Now, apart from that, you can also have hydrophobic interaction. Remember van der Waals? Okay, different forces. So what happens is between these non-polar amino acids also there will be van der Waals force or hydrophobic interaction. So you can see all different kinds of interaction in this tertiary structure. You have ionic bond between positive and negative charges. You have disulfide bond between two sulfhydryl groups. You have polar forces, okay, between partial positive and partial negative atoms, okay. So they will be forming the hydrogen bonds. And then you can have non-polar interactions. Okay? Non-polar interactions will be between non-polar amino acids. After tertiary structure, you have the final stage. This is seen only in some proteins. It is called quaternary structure. Quaternary structure is when many polypeptides, okay, many such tertiary structured polypeptides or proteins, they come together and they form a complex. Okay, example is hemoglobin. Just look at this diagram here. Okay, so you have, this is the primary structure, which is nothing but a chain of amino acids. This chain of amino acid gets converted into secondary structure, which is alpha helix, okay, or it can be beta sheet like this. And this alpha helix and beta sheets, you can see they are finally folded, okay, into this tertiary structure, which is ball like, right? It is ball shaped. It has this alpha sheet or alpha helix also. It might have beta sheets. It will have other turns and twists also. Now with this tertiary structure, you can see something different. This molecule is nothing but heme group. Okay, heme is like porphyrin ring. Okay, have you seen the structure of chlorophyll? Right, so that's chlorophyll. In between, what it has is nothing but a porphyrin ring. Okay, so same way porphyrin ring with iron in center, Fe. So you have Fe in center, and then you have this kind of a ring structure. I'm not able to draw it very well, but I hope you will be able to understand. Okay, it will have this kind of a rings like structure and like this. This does not look very good, but this kind of a structure is called heme, okay, because it has Fe, iron in the center. So along with this tertiary structure of protein, you can also have heme group, okay, and when four such tertiary structured proteins, they come together, they form a hemoglobin. For your information, here you can see the 4, 1, 2, 3 and 4. Hemoglobin is made up of two alpha type of chains, okay, alpha polypeptides or two alpha type of proteins, and it has two beta type of proteins. Clear? Remember this, it has two alpha chains, two beta chains, because this was asked in 1 year, okay, in one exam. Also, when you will be studying genetics, this will be important. So remember, 2 alpha, 2 beta. Summarizing the whole lecture, Two amino acids are joined by condensation. Condensation is removal of water. And a peptide bond is formed. Peptide bond is CONH. Protein structure is divided into 4 levels. You have primary structure, which only has peptide bonds. Okay, it is a linear polypeptide, a straight chain. Secondary structure has peptide bond as well as hydrogen bonds. Okay, and with help of these hydrogen bonds, you are forming the alpha helix and the beta sheets. Then you have tertiary structure. Tertiary structure is the one where there is peptide bond, there is disulfide bond, ionic bond, hydrogen bonds, as well as hydrophobic interactions. And it has a 3D structure. So remember tertiary and 3D. Tert means 3. So 3 and 3. Quaternary is when you have multiple polypeptides joined together. Living state is possible by input of energy by metabolism to do work. This we will be studying in the next class. Fine. So I hope you had fun and this was something new or some better way to understand amino acids as well as proteins. Now your another homework will be to go through your textbook and make a table of all different proteins mentioned in your textbook. Okay, as many as you can, not wasting a lot of time, but just write down as many proteins as you can find in your textbook. Okay, just to see that how much diverse it is. Clear? Okay, see you next time.